Okay, I think we can go ahead and get started if everyone is ready. I'm going to just start with the brief introduction and then we will get started with the presentation. So thank you. Happy August. Thank you for being here for our monthly research seminar series. Today we are hosting Dr. Natalia Quinete. She has a Bachelor of Science in Chemical Engineering, a Master of Science degree in Analytical Chemistry, and a PhD in environmental analytical chemistry from Brazil. Since spring 2020, she has been an assistant professor at Florida International University, FIU, in the Department of Chemistry and Biochemistry, and is affiliated with the Preeminent Institute of Environment at FIU. Her research focuses on assessing and understanding the sources, distribution, and fate of persistent emerging contaminants such as PFAS in different environmental and biological compartments and their potential impacts on humans and animals. More specifically, she has been working on the development of targeted and non-targeted analysis methods using high-resolution mass spectrometry for the identification and prioritization of contaminants of concern in the environment. Today, she'll be sharing a presentation titled Environmental Fate and Human Health Implications of PFAS in South Florida, an integrated assessment of atmospheric, aquatic, and biological matrices. Without further ado, thank you.
Thank you, McKenna, for the nice introduction. Actually, since a month ago, associate professor. So
Good to
yes,
know. Thank you.
yes, I have been gone. Yeah, just got tenure. Thank you so much. Yeah, today I'm going to be presenting a little bit about some of the studies, research that we have been conducting in my lab. So that are mainly focused in South Florida. So now, okay, go. So I think most of you have seen that lately there is a lot about PFAS. I imagine that there is a lot of people that already know what that means. But for those that you never heard about it, PFAS stands for per and polyfluoroalkyl substances. So these compounds actually, they have been discovered and synthesized over 6 years ago. And as you can see in this figure here, they have been using for quite a wide variety of both commercial, household, industrial products, such as like AFFF, so firefighting foams, food packaging. They are also present in formulation from pesticides. They are in non-stick, like very famous Teflon, right? In non-stick pens, clothing in. So, and as you can see here, in a huge variety of products. And this is one of the reasons there are so widespread presence in the environment and also in humans. You probably, if you follow, have been looking at the news. There has been quite an increasing in reports on PFAS in drinking water or in all sort of environmental matrix. A while back, I always tell my students about this movie, Dark Waters. So PFAS is so famous that it even made it to Hollywood. So this is a story about the lawyer, Robert Below, who fighted DuPont and their illegal disposals from waste containing PFAS that were contaminated and causing a lot of cancer in a community in Virginia. And I mean, not only, the only problem is that besides PFAS, PFAS actually made it everywhere. So this map here is just showing it, all of the occurrences of PFAS in different locations drinking water. The truth is that you would have a lot more blue points over here, but there's still, as you can see, a lot of locations that have never been assessed for PFAS. So that's why you see like more concentrated in some points where there's research going on. But the reality is that it's quite rare that you analyze a sample where you don't see any PFAS. So for those of you that have never seen the molecule of, this is just a structure. How do they look like, right? Like their template. So you can see their carbon fluoride alkyl chain that is very representative from all the PFAS. There kind of gives them all those hydrophobic properties. And at the same time, they have like a functional group which is their smaller head, that are extremely hydrophilic. So that's why they make them so good as surfactants, right? Like they are able to both dissolve, but also repel water and oil. So, and some of them you can see here, so those carboxy group, you can see they can change it, right? Like you have, and there is what actually they differ in different classes of PFAS. So we have the carboxy group, one of the most, I don't know if I'm putting here, maybe I should put some laser pointers, sorry. So you have here the PFO8, which is one of the most predominant from this group. And then you have on the sulfonic group, you have PFOS. That is one of the most predominant PFAS from this group found in environmental samples. And it's always biological samples. So those chemicals actually different from the our traditional chemicals like PCBs. I'm pretty sure that everybody have heard about it. They can accumulate it in fat, but also they have a preferential binding into proteins. So, and they have been shown to have a wide variety of adverse health effects. So this is, I always like to show this graph. It kind of shows you some of the things that have been already associated with PFAS exposure. And, you know, the other thing here that is in bold, it shows actually the ones that have more evidence, so more clinical and epidemiological evidence. So they have been caused delayed in development, reduced response from vaccines, associated with thyroid diseases, increase the cholesterol levels, increase of obesity, and liver damage, and also have been potential carcinogenic. There has been shown some links to potential cancer types, like kidney and testicular cancer. If you look at the sources, and you can see the majority of those sources we have here in Florida, right? Like, so those PFAS could be coming from firefighting foams. They're used in airports. They're used in air bases, military air bases, and you know, all any like firefighter training places that it can eventually lead it to leach out into the surface water. They are being, of course, used in some in the production of the most common household items and food packaging. So they have been also linked to use in industry. We don't really have a manufacturing Teflon here in Florida, which is a good thing. But there are other companies that could be being using it, or at least in the manufacture of products that use this PFAS. We can have an input from wastewater, either for accidental discharge, can also lead to PFAS going to surface water. We also, from all of their uses at home, they actually can end up also going into landfills where most of the waste goes. Also from the wastewater, some of the sludge go to landfills. Biosolids can be used in agriculture. So again, all of the sources can end up of either accumulation here, for example, in soil can accumulate in crops, or it can end up runoffs and end up in the surface water and then eventually infiltrating groundwater and then on our drinking water. So most of our water treatment actually are not well equipped yet, or they have been not designed to remove those chemicals. There is something that is eventually facilities are working towards, but again, this is one of the ways just to explain how they end up in the water. They can actually also accumulate it, and some study has shown that they tend to biomagnify as well. So not only accumulated in our food supply, you know, fish and crops, but also they can biomagnify through the food web. So usually top predators have the higher concentration, which include us, right? So some of the most known human exposure pathway, again, this is not comprehensive. Of course, there is some study that might be also being through dermal, but one of the major sources through drinking contaminated water, eating food that has concentrations of PFAS, but also I'm going to show you guys some data from how you can also be present in the air. So regulations on PFAS is always an evolving topic. I don't know if many of you heard, but this was a sum of the maximum contamination level, and those are the list of regulated PFAS that were established in 2024. Very recently, this data is being revised to only PFOA and PFOS, so the other PFAS going to be some, drawback from the other PFAS. But I'm just going to show you some of the data that we got, and we have been analyzing a lot more than only those regulated PFAS. And there also a hazard index that was calculated based on the synergetic or addiction effects of having more than one PFAS in the drinking water. So based on those maximum concentration levels, the facilities, all the water facility have been currently monitoring those PFAS in the water. And the goal is that they have a couple of years to be monitoring, and if concentration exceed, there is a need for new infrastructure that is able then to treat it, especially PFOA. PFOS, you're going to see that most of them usually exceed the regulations, but there is a need of infrastructure to treatments to be able to reduce those levels from drinking water to the acceptable levels. And again, if that is not achieved, then they're going to be a need to find alternative, which are probably no water facility wants that. So they're currently working on monitoring and trying to develop a test that can remove those chemicals. So what we know so far is when I got here at FIU back in 2020, PFAS in Miami, there was it was actually have that news that PFAS in Miami is one of the highest from the different states and the different locations that were assessed in different states. Actually, I think it came out as a third highest from 44 different states. And it was about the same time I started at FIU. So even though like my story with PFAS is a little bit longer, I started working during my PhD in 2006. When I saw that, and I said like, okay, there's definitely a need on some PFAS studies. So we did a review paper on trying to look what we knew so far in Florida. And there was very few studies. So this map here is like there were very few studies that had assessed. Again, there's not very comprehensive. This is a study that has been done in either sediments back then, groundwater. Most of the places you can see they were closer to military bases sites. And one of the things that we saw is there was really high levels in Miami-Dade. So we definitely thought like there's a huge need of more research on trying to better understand, right? Like what are the sources, what is the levels, what is the distribution of PFAS here? And again, and back then there's really not much about what is the fate of more emerging ones. So PFOA and PFOS we know has been banned, and then some other shorter chain alternative and have been replaced, but not much it was known about their fate in the environment. So overall, there was a need to try to understand how those chemicals enter the environment and what are the risks associated with both environment and human health. So most of the research in my group has been focused on trying to assess the fate and source and environmental and human impact for those emerging contaminants of concern. A lot of the effort has been put now on PFAS, but we also have been looking at plasticizers, disinfecting byproducts. We have been focusing also in improving analytical methods, generating more greener alternative, more high throughput methods for measuring contaminants of concern. and use it of, and I'm going to be presenting also here today, so use of specific biological species as bioindicator of exposure to assess local contamination, or also human biomonitoring to try to understand what are the potential human exposure risk and health effects. So when we started, one of the first works we were doing is trying to understand both surface water and tap water concentration of PFAS. So there was actually no work here except for one sample that was collected. It was one of the highest that I mentioned before. So we, of course, for the proximity of Biscayne Bay and the importance of Biscayne Bay is from the area, we started collecting samples from several of the canals that discharge in the bay. We also collected from Everglades canals. Again, both Biscayne Bay and Everglades aquifers are the main sources of drinking water supply. And to compare that, to have a better idea, we collected samples also from Tampa Bay. We had some samples that were collected in more saline marine environment in Key West, so there's more saline water. And we collected tap water mainly from the Miami-Dade, Broward, and Palm Beach. But we extended, and we had like some small amount of samples that we collected from different counties as well. So there's a work from one of my graduate students that she graduated in fall 2022. So we developed a method where we're using 250 milliliters of the water. We concentrated this water, right? Like the levels are usually very low part per trillion levels. So we concentrated in solid phase extraction 250 times and to 1 milliliter. And then we were analyzing in a liquid chromatography mass spectrometer, right? A triple quadrupole mass spectrometry. We started initially with a list of 30 PFAS, and actually nowadays we have been expanding it. I'm going to show you some data where we expanded to analysis of 40 PFAS, the more newer method from the 6033 from the EPA. So in the initial assessment that we did for 30 PFAS, we actually this is the data that we saw in the different areas in Florida. So we can see that among the most predominant PFAS, we have PFBA, PFOS. So even though it has been restricted, it's still one of the major chemicals that we see in drinking water. PFPEA and 6,2-FTS is one of the major compounds associated now with firefighting foams, since PFOS has been not in use anymore for the older formulations. So we saw that in terms of the sum of PFAS, it could range all the way from 1 to 230 nanograms per liter. And I think is evident here that the highest concentrations we actually saw in the East Coast of Florida compared to West Coast and in Central Florida. We also decided to expand on this collection, and we tried to integrate it a little bit more environmental health aspects. So we prioritize sampling it based on liver cancer mortality. So basically, if you look at here in terms of cancer counts or mortality in Florida, we see, of course, like the top one is breast cancer, and then prostate cancer second, and third one is liver. So breast cancer and prostate cancer, there was already some evidence that PFAS could be linked. There's already like high evidence from that. And for liver, we know that PFAS could cause liver damage, but there is not anything supportive of liver cancer. So we separated in different quartiles, both collections. We collected both in Palm Beach and so Miami-Dade. So the first quartile is areas where it has lower mortality. And then the fourth quartile being the one that has the highest mortalities for cancer. So based on that, we have collected about 72 samples of drinking water in Miami-Dade, 75 in Palm Beach. And you can see we tried to collect it, enough samples from, so we collect from 18 zip codes in Miami-Dade, 22 in Palm Beach, and we try to collected some good amount in the different types of quartile, especially like the 3 and 4 representing the higher cancer, and the 1 and 2 represented the lower cancer mortality index. So this is the data that we got. So it was very interesting to see that we have quite a different profile from PFAS. So Miami-Dade, we saw a lot more contributions, for example, PFBA, 6 to FTS, most likely probably because of the airports and the airbase. Then PFHXA was also could be coming also from the AFFF foam, and as well as we see PFOS. In fact, if you see like all the green ones here, Miami-Dade concentration is a lot higher than Palm Beach. And in Palm Beach, you can see that majority was PFOS, but you have also some contribution from PFBS and PFHXS. So you can see here, according to the maximum concentration level from PFOA and PFOS, you can see that, many of those locations in Miami-Dade is exceeding it, but not for Palm Beach. So this is, by the way, this is the average concentration. So I'm going to show another graph next showing it. Comparison about exceedance in the 2 counties. So here, we see that when we try to plot, like in terms of the harder index that as I said before, like his contribution for different, more than 1 PFAS in the drinking water, we can see that in Palm Beach, none of the zip codes it says actually exceeded this hazard index criteria, but we saw 2 of the zip codes, and some of them even reaching out pretty close to this hazard index of 1, 2 of them exceeding it. So looking again at the spatial variation, you can see that, again, it's very clear that concentrations in Miami is a lot higher than in Palm Beach. We saw about 56% of PFOA exceeding Miami-Dade. 99% of the samples exceed the criteria for PPT for PFOS. And in Palm Beach, we have 20% exceeding for PFOS and only 3% exceeding for PFOA. Again, the problem about the exceedance, especially in terms of PFOA and PFOS, is that this could lead to potential health outcomes. So there's something that is very important for us to be monitoring those levels. Again, we look at the different quartiles and we try to see what is the concentration of PFAS, the sum of PFAS. So this is plotting here. Quartile is our proxy for liver cancer mortality, and the sum of PFAS. I think is interesting to see, like, we didn't find any statistical difference between the quartiles when doing Kruskal-Collins test. We're now looking at other statistical tests as well, but we can see clearly that even though the medians are different, but it looks like that, at least for quartile 4 for Miami-Dade, it looks concentrations higher than 1 and 2 and 3. And then here for Palm Beach, we can see that quartile 3 especially sounds to have like a higher concentration, even though not statistical different, but higher concentration than the other ones. And when we look at for individual PFAS, I'm not showing the graph here, but we did see that some of them, both the Miami-Dade as well in Palm Beach, show a higher, for example, PFOS show a higher concentration in the 3 and 4 quartile and a higher mortality quartile than compared to the 1 and 2. Okay. So when we look at the surface water concentrations, what we can see is that Biscayne Bay shows overall the highest concentration of PFAS compared to the other water sources. Of course, like I'm going to explain a little bit later about this. Like this is clearly an outlier, But overall, if you look at concentrations are higher in Biscayne Bay, more specifically have identified some of the canals that has a higher input of PFAS into the bay, which was Biscayne Canal, Little River Canal, and Miami River Canal. And after that, we have Tampa Bay showing the highest concentration, followed by the Everglades, and then the smaller concentration, Key West. So about this one, this position, this collection site that calls HB, actually is the same canal as the CO3 here. So this canal starts here, the Everglades, and then it flows all the way, passing about 2 kilometers away from Homestead Air Base. So that's the closest canal to the base, and that's where we identify. So we are assuming that most likely this higher input here in this point from this canal is likely related to the this former military air base, now is a reserve base. So we expanded then. So when we identified those major canal in Biscayne Bay, so we expanded and trying to identify what could be the potential pollution hotspots, right? So we collected samples along these 3 canals, and also in collaboration with Mikusuka tribe. They were very concerned about what would be the background levels of PFAS in the community. So we collected in both canals and marshes, and along with the water resources team there, director there. And again, what we saw is the levels in these areas, they're clearly lower than in the major canals that receives a lot more anthropogenic influence. So we saw here that Miami River is the one that has the highest input. And it was interesting to see that we usually observe, like as you can see also here from Biscayne Canal and Little River, is that the concentration are increasing as they approach the mouth of the bay. And also that the majority of those areas that had higher hotspots, like the higher levels, were associated near usually airports, hospital system, wastewater areas. So we don't really have like a regulated level, but Florida DEP, they have some screening level for both PFOA and PFOS. We found that about like 43% of our samples exceeded the surface water screening levels for PFOS, which is the 10 nanograms per liter, but for PFOA, we didn't find anything exceeding it, which the surface water screening level was about 500 nanograms per liter. So I just wanted to show you, so this is in orange are our sampling points, and everything here in blue is a sum of the potential sources. So you can see here that, some waste and sewage facilities are closer, departments are close by. We also have input from firefighting foams, from airports, fire training departments. And we also see, some industry that could be contributing, like a boating, vehicle repair, metal recycling, constructions, as well as a golf course that is closer to Biscayne Canal that could be also contributing. So we have different types of PFAS sources. So going more deeper in Biscayne Bay, we want to, so PFAS are getting to the bay, but how are they behaving? And once they arrive, our marine environment. So we collected samples, like, you can see my students, graduate student, undergrad student, they collected different waters from both the surface, and now we also wanted to see what is the profile, not only vertical, but also horizontal. So what we did, we collected surface and deep water samples from different transects that we did here in the bay, all the way coming from the major canals until offshore. And what we saw is usually a decline in PFAS concentration. So even though those concentrations are higher, closer to the to the coastal area, you can see that the concentration are usually, as you can see, declining as they approach offshore locations. See, offshore location usually has the lower concentration. And in terms of what it was the concentration in terms of surface and the deep water, what we saw is in surface water, our concentrations usually higher than in the deeper water. So deeper water, again, remember I'm in Biscayne Bay, I think the deepest that we have is about 4 meters. So it's not really that deep, but you usually have some depletion as concentrations go down, which makes sense because most likely what is happening is some of those PFAS are actually absorbing into the sediment and precipitating it, right? So this is one of the next goals that we want now have to see, like what is the profile in the sediment core. So my student also apply inverse distance inverse distance weight model. So, and to forecast or try to see how those PFAS are dissipating. It's kind of mimic, and it was very interesting. We could see that the northern section clearly has the highest PFAS, so red is the highest. And in the south area, you can see yellow, so concentration is usually a little bit lower. And offshore, as you can see, a green. So this is where you have the lower concentration. So he kind of plotted how PFAS would be distributed in the water system. So if we have them in the surface water, how does it look like for our groundwater? Well, so what is the impact from that, right? Like there's a lot of our drinking water coming from Biscayne Bay aquifers, right? So what we did is in collaboration with Miami-Dade Water and Sewage Department, we collected some of the water, groundwater from monitoring wells. And this is the data, as you can see here, you know, again, PFOS, all concentrations exceeding it, the maximum concentration level, PFOA for some of them also exceeding it. The highest concentration groundwater we found was for PFHXA. And when we applied the hazard index, we saw the Miami Spring, so one of those wells is the only one that actually was exceeding it, the regulations. And in fact, this well has we heard that this well actually is not being in use anymore. So this well has been closed because of the higher PFAS levels. So again, if this kind of shows us like all the concentration, all the levels of groundwater are ending up in a drinking water, which we saw a very similar profile with PFOA and PFOS. So we know also that these chemicals are very stable. So because of their stability, one of the things is that they can actually travel long distance. Even they have been detected in the Arctic and Antarctica and areas very remote and very far away from any production sites. So, and the reason for that is that they can be transported either through atmospheric current as well as oceanic current, and then can end up in these areas. So for that, we kind of like, even though again, we have some different sources, but we wanted to understand what is the input? Does it really non-point sources like atmospheric deposition really matter here? So we had a study, we conducted 1 year collection of rainwater, and it was very interesting. So we saw like in the different months, we calculated concentration of PFAS, we're able to estimate what is the flux of PFAS. But more interesting is we applied this data into look it up and see what is the contribution from the air masses. So we calculated back trajectories. So it was very interesting to see that the rainwaters that has the highest PFAS actually, you know, we had a moderate influence. The majority of the influence was coming from northwestern air masses. And then the lowest concentration of PFAS is coming in from the southeast air masses. Again, we all know that in the areas here in the north, you know, like you have North Carolina, and then you have, you know, here other industries that have PFAS. So some of those PFAS, actually we detected GenX, that is one compound that are associated with manufacturing of PFAS that we have not detected in water here, but we did detect them in rainwater. So what about ambient air, right? Like we look at the wet deposition, and we decided to do also have a look in the dry deposition. So these are just some examples from different sources for inhalation sources. So we can come from household dust, it could come from pollution, right? For industrial pollution, PM 2.5, can be used for cleaning product, food packaging, cosmetics, electronics, contaminated soil. And again, my student has set up also to collect, ambient air, so atmospheric particulate matter. And we set up here a sampler here at BBC in Biscayne Bay. So it looks something like that. I have a little picture. So basically the sampler, as you can see, it has different stage. So the particles here, each one of them has a filter. So the bigger particles get stuck in the first filters and then go smaller and smaller. And then, the sixth stage is going to be the smaller particle level. So, and it kind of mimic our respiratory tract, as you can see here, where you have like the higher particle size, we're going to get stuck here in the nasal cavity, but then the smaller as it goes in the last stage, it can actually reach out of the alveoli and enter eventually the bloodstream. So we collected samples for 48 hours a month, and for a whole year, and we use in each of those stages here, we use a glass fiber filter to then collected our atmospheric particulate matter. So this is the data that we have for PFAS, so concentrations in picogram per cubic meter. So we found that, you see, some among the highest concentration we found for the most volatile PFAS, like PFBA, and also PFPA. And then also we have contributions from PFHXA, right? But besides those ones that has a high abundance, we also found some other PFAS were highly detected. So that has almost 100% detection rate, or not 100%, sorry, I think, it was about 30% or to 50% detection rates, like PFOS. We saw PFOA and also methylphosphamide. And besides the PFBA and PFHXA. So some of the potential sources from that, of course, we have influence from indoors, right? Like we are in an area that has high traffic, open the doors. It can also have contributed. We have African dust, especially during summer, that could be contributing to bring a lot of particulate matter. We have close by a power generation plant. We have incinerators. Road dust could have also contributed to PFAS level, or even sea spray aerosols could have contributed to the input of PFAS into the air. So we calculated here a distribution for the particle bond. So this is a little bit complex. I can explain it later, but we kind of normalized the size distribution, the different being, and we were able to calculate it. We did have calculated logarithmic size interval for particles collected at each stage. So we basically, based on the aerodynamic diameter. So here it was very interesting to see that some of PFAS, so like PFHXA, PFBA, and methylphosphamide were more usually deposited into the coarse particles, so above 2.1 micrometer. And then we saw that PFOA, PFOS, and PFPA, they're usually more associated with fine particles below the 2.1 micrometers. And it's important to note that there's actually currently no regulation of PFAS in ambient air. So there's something that definitely more studies are needed to be able to establish that. We also calculated the deposition efficiency. So how much is deposited into the different stages, of the respiratory tract. So we saw that about 2 to 30% were deposited in a alveolar deposition. So that's the one that can eventually get on into our bloodstream. About 1 to 6% is in the tracheobronchial deposition. And then in the head airway, so upper then, we saw that's the most particulated were about 60 to 90, 60% to 96%. We also calculated what is the daily exposure based on a 24 hours per day exposure. Of course, we always breathing in. So we try to calculate inhalation health risks based on, again, these are only based on what we have the oral minimal risk level. So there we have a reference dose just for PFOS, PFOA, PFNA, and PFHXS. And you can see here, like, we calculated the risk based on either the coarse or fine particulated, and we calculated for three different groups, so males, females, and children. You can see that all of them hazard index is below 1. So this would usually be related to a low risk level. But again, there is still several limitations, especially because there are just a few chemicals are being in. We have actually toxicological data for a very few chemicals, so there's still a need for more information on other PFAS to be able to better assess what are the cumulative inhalation health risk. So changing a little bit here to looking now in biological samples. So wildlife has been used for long as a bioindicators to assess environmental contamination, also to assess overall health of the ecosystem. So we are studying more specific oysters. So oysters have filter feeding bivalves, and usually we use them as a bioindicator of local contamination, especially because they have higher capacity of bioaccumulating chemicals. They don't move, right? Like fish, they usually move, but oysters, they have a specific location, they limit movement, and they usually can find broadly. So you can actually also use to compare between different locations. And again, we have also developed a method for oyster extraction. So we collect a total of 156 American oysters in 3 different locations. We collected in South Eastern area. So it was here, actually at FIU in our marina. And we also collected in southwestern area in Marco Island and in central east area in Terracea. This actually is an oyster farm. So we collect in all those locations. We develop a method based on ultrasound and a cleanup with fluorizil and graphitized carbon black and analysis by the same instrument, LCMS-MS. So what did we find out? So we found, again, PFBA, PFOS, and this longer chain PFTEDA was the major chemicals found. But overall, we found PFAS in all of the oysters that we assess, okay? And you can see by the graph that Biscayne Bay seems to be the one that shows the highest concentration compared to the other locations. So to confirm that, we did do a Kruskal-Wallis test, and we found out that Biscayne Bay was indeed statistically different from the other side, so having the highest input. And we also calculated condition index of the oysters that are based on the oyster thickness, height, and width. And the goal of that is try to assess their health. And we did some experiment correlation to try to see if PFAS has correlation with any of the parameters assessed here from the oysters. And it was very interesting to see that there were, so some of PFAS was negatively correlated to the condition index 1, as well as animal weight, shell thickness, and the reality is that Biscayne Bay has the smallest oysters and the ones that has the high and the less thick shells, and is the one that has the highest concentration. So we, again, is not anything causative, but it is an indication that maybe, PFAS could be, along with other chemicals that might be present, one of the contributors to, oysters impact, adverse impact that we observed. We also expanded to collect also fish and some invertebrates. So we try to get recreational fisheries that are most consuming for Florida. So we collecting black tuna and lobster. We also collected red drum. So red drum is also one of the top 5 most targeted recreational fisheries in Florida. And this is a very comprehensive study that was done in collaboration with another faculty here, Jennifer Rehash, where we collected red drum from different estuaries here in Florida. Red drum also, I don't know if you guys know, but it's very famously used for this recipe for this chef, Paul Prudhomme, I think that's how we pronounce Prudhomme. So again, we looking at the data first for the fish, for the tuna and lobster. So again, they are very distinct. So fish is a vertebrate, lobster invertebrate organism, and they also have very distinct habitats. So lobster are more benthic organisms, where tuna are pelagic organisms. And it was very interesting that we saw very distinct distribution. So the benthic organisms are closer to sediments. So we saw a lot more contribution for longer chain that usually are present, absorbed into sediments, so higher concentrations. And then in pelagic, we kind of saw similar, we have some also high contribution for long chains, which again tends to accumulate a lot in fish and in biota and biological samples in general. And we also saw PFOAs as one of the major contributions, what we didn't see in lobster. For red drum, it was very interesting. Again, red drums are actually also benthic fish, so it's different from tuna that is more in the surface. So red drum is more closer to the sediments. So again, we also saw the contribution of methylphosa, which is one of the PFAS precursor that we also saw in lobster. But PFOS is one of the major contribution as well, and as well as PFOA. You can see that PFBA. So PFBA also has one of the highest concentrations found in the fish. So overall, PFAS were found in any other fish in the estuaries. And then it was very interesting to see. Again, we didn't assess, we assessed Florida Bay, but we didn't assess anything here in Biscayne Bay. But we can see that for Tampa Bay, along those all different locations, was the one that has the highest impact, so that has the highest concentration in fish. We also, from the data that we have from Biscayne Bay, we had the data from the water, we have the data from the fish. So we actually were able to estimate bioaccumulation factors. So from the bioaccumulation factors, you can see that those PFAS are accumulated. The highest one we saw is a C9 PFAS, or is a long chain PFAS in lobster, but it was very interesting to see the shorter chain PFAS will also have being able to accumulate in both in lobster and in tuna fish. And we saw that in terms of the different classes, the fluorotelomero sulfonates are the ones that are accumulating the most, followed by the sulfonates, and the carboxylated, the one that are least accumulated. Again, they're usually more present in the water. They're usually more soluble, and they have higher solubility. That's one they usually accumulated more in the water than in biological sample. So we calculated some human health risks. Again, similar to what we did for the air, but of course, different calculations here. So we calculated estimated daily intake, and we calculated a hazard risk for only for the PFAS that we have, the oral maximum level available. So we did see that all of them, we calculated for consumers or normal consumers, and for heavy consumers, so those ones that consume a lot of seafood. And we saw again the hazard risk is below 1, so lower risk. But in this case here, we actually try to calculate it just to estimate it, how much fish or how much oysters or how much lobster can you eat, I mean, like for try to drive a recommendation letter, recommendation guidance, right? Like, so if you, for fish advisory level, so if you eat more than 13 meals of tuna, then you probably will have level that would represent the risk. If you eat more than 59 meals of lobster, then, you also would represent the risk. So we try to do that. And for oyster, you end up being 579 oysters per month. And again, I don't think anybody eats that much oysters a month. They're very expensive, but again, this is just an idea for us to know, like, how much will people have to actually eat to be maybe affected, by PFAS. So that could really represent a health risk. Again, there's only 4 PFAS. We were measuring 30 PFAS in total. There's still a lot of, need for toxicological data on other PFAS to be able to better derive health risks. We also had a project. I mean, I'm going to show really quick. I know that I'm running out of time. So this is just, we had a collaboration with SeaWorld scientists, and this is one of my postdocs that work with me. And she measured PFAS in 2 different types of dolphins. And what we can see is, again, she did in serum samples. You can see concentrations wise is a lot higher. So we found here from bottle dolphin, actually concentrations were higher than in orca. And it was very interesting. We have both mother and calf pairs. And what we saw is we were able to see PFAS transfer via placenta. So you can see here, this is the mother. So after giving birth to the calf, you can see the concentrations decrease, both here too, like you see concentrations are decreasing it. And then concentration in the calf, we actually got the samples from the same individual over several years, okay? So here you can see the concentration is increasing it. So again, this is through lactation, and you can see that this period here was marked is the weaning period, so where the calf stopped lactating, and you can see how concentration of PFAS start decreasing after that. So it was very interesting to see this contribution for both placenta and lactation in this study. I just wanted to point it out that we have a lot of other projects going on. Again, besides trying to understand the fate and identify the sources, we also being involved in several projects that are trying to investigate and evaluate different treatments. So we have this project here with the University of Cincinnati where we're looking for ultrasonic degradation of PFAS. We have a project here, here at FIU in collaboration with PNNL, where we're trying to look for developing new MOFs for remediation of PFAS in groundwater. We have another study that we're looking also for degradation of PFAS using electron beam. There's a collaboration with Jefferson and Fermi Labs. And also we have another one, a collaboration with the University of Miami, where we're developing some high entropy alloys for mineralization, and we're testing a different type of recalcitrant chemicals, including also PFAS. Again, just some take-home message. I think we have seen the Florida coast, including Biscayne Bay, fisheries and wildlife are being exposed to PFAS. We have identified some of the major sources, which I think they're kind of expected, like airports, military airbase, effluents from wastewater treatment plants, and also the influence from anthropogenic and domestic input from the canals. We have identified some of the predominant PFAS that are present in tap, surface water. We also have done some work on biosolids and wastewater. We also have seen these chemicals are present in rainwater, air ambient, ambient air, sorry, and then marine life. But again, there's still, I hope that this was evident, there's still a huge lack of toxicological data. So there is still, that we need that to be able to proper assess health risk. And again, the majority of the treatments that we have are not really efficient in removing PFAS. I know that the facilities are working through, I think they're implementing an GAC, so graphitized activated carbon, as well as they're looking at into other ion exchange for removal of PFAS from drinking water. Again, we also been looking for different types of solution that can be effective. Besides what I mean, I show most of the data on target analysis, but we also have been developing method and assessing it, PFAS through non-target analysis method, so which is capable of actually more comprehensive assess the amount of different PFAS that we don't even have the standards for. And moving forward, we also looking to be working in evaluating the fate and transformation of volatile PFAS. So we currently developing method for volatile PFAS. So to try to understand their fate in the atmosphere. And also I have been collaborating to try to assess toxicity of those emerging, especially for the emerging PFAS. There's still a lot of data, PFOA, PFOS, but very little about the most emerging PFAS. I like to, of course, acknowledge all this research would not be done without all my students, postdoc, all the amazing collaborators, the Institute of Environment here at FIU that gives me a lot of support for my research, and all the funding agencies that has supported my research. And again, this is all my amazing group of students. Some of them already graduated. Some of them are still here with me. And thank you so much for attention, and I'm happy to answer any questions. I see from the chat there is a lot.